Hello, welcome back. Uh, this is going to be first of hopefully many lectures in a series dedicated to cofactors, which are used up by different cellular enzymes. Today, I'm going to be talking about nicotinamide adenine dinucleotide. Let's just unravel the name itself based on the structural attributes of this wonderful cofactor, which assists lot many enzymes catalyze redox reactions. There are two varieties of this molecule existing in cell. One is the unphosphorylated form, which typically what we refer to as nicotinamide adenine dinucleotide. And then there is one more variety which is phosphorylated NAD. Therefore, it's called as nicotinamide adenine dinucleotide. Okay, so these are cofactors which are used for redox reactions, although in different paradigms. What we'll do is we'll take a look at the structure of these things, and then we'll try to understand how these molecules are involved in redox reactions by taking one example. I think that should be simple enough, using lactate dehydrogenase catalyzed conversion of pyruvate to lactate, or the other way around also, okay, because it's a reversible enzyme. But before we get into the mechanisms of redox reactions in which NAD or NADP participate, let's just take a look at the structure itself. Okay, what you're looking at here is the structure of NAD as well as NADP. Now let's just understand why it's called as nicotinamide. Let's just a minute. I think I moved my slide. Okay, let's just understand why it's called as nicotinamide adenine dinucleotide in the first place. You have adenine moiety here. I'm not going to explain the structure of adenine or any other groups in this video. But if you'd like me to explain the chemistries of these, let me know. Or I think they're pretty simple enough. You can figure out it by yourself. But you have an adenine moiety here, and then a ribose attached to it. So you have adenosine and a phosphate. So this together is one of the two nucleotides which are present in it. Okay, so you have the AMP here, and then the AMP is attached to one more nucleotide. You have pyridine here. Of course, the pyridine is in the cationic form here. The nitrogen is in cationic form. Pyridine in turn is attached to a functional group of the type of amide group. And then you have a ribose attached to the nitrogen. And then the ribose is also attached to a phosphoryl group at the fifth carbon atom. So these two phosphoryl groups, one of the this part of the molecule and one of the AMP, are in turn attached to each other by a phosphoanhydride linkage. And that's why it's called as nicotinamide adenine dinucleotide, simply standing for you have nicotinamide here, you have AMP here, and nicotinamide is a part of the nucleotide as well, okay, because you have nitrogenous base, a sugar, and a phosphate. Here there is one more nitrogenous base in the form of adenine, sugar, as well as a phosphate group. These two nucleotides are in turn attached to each other by a phosphoanhydride linkage. Okay, this one. So it's called as NAD. That's one thing you got to keep in mind. The second thing I'd like you to appreciate is NAD is often written as, I mean, you must have seen a lot of textbooks or papers or whatever. NAD is written as NAD plus. Why is it like that? Because it's in the cationic form. In the oxidized form, okay, if you can pay attention here, the nitrogen, the pyridine moiety is in the cationic form. That's simply because the lone pair of electrons of the nitrogen are used up to make another bond, covalent bond. You have 1, 2 of this double bond. So that makes up for 2 covalent bonds. Third one is here, fourth one is here. So together, I mean, there are 4 covalent linkages. And therefore, the fact that the lone pair of nitrogens, which otherwise would be present on this nitrogen, are being used up to form a covalent bond, and therefore nitrogen is electron deficient, and that's why it's in the cationic form. And that's the reason why NAD is depicted as NAD plus. It's as simple as that, because it's in the cationic form. And please keep in mind this cationic form of the nitrogen, because it will come into considerations when we talk about redox reactions. Now, what we'll do is we'll just move over to the structure of NADP. Okay. Now, the only difference between NADP and NAD is that the second carbon atom on the ribose moiety attached to adenine is phosphorylated in NADP, as you can see here. Okay, that's it, nothing else. it really has no bearing as such on redox reactions. It's just that NAD plus versus NADP plus are used by redox enzymes, no doubt, but which operate in different paradigms. If you look at metabolism itself, okay, you will see that NAD plus is used as a cofactor in catabolic reactions, where the electrons are invariably removed and given to, one second, and given to NAD, as NAD becomes NADH, okay, NADH, right? And in most cases, the fate of NADH is by direct or indirect mechanism, it's going to be oxidized by electron transport chain. So that's the catabolic reactions in which NAD is used up. Unlike that, NADP is used up in anabolic reactions. Okay, so NADPH, which is the reduced form of it, comes into the reaction and gives 2 electrons for the anabolic steps involved in reductive anabolic reactions. Think about cholesterol biosynthesis or whatever, right? Or free fat, or the activity of fatty acid, fatty acid synthase. In all those reactions, NADPH is used, and principally NADPH comes from pentose phosphate pathway. That's the importance of pentose phosphate pathway, but I'm not going to go into that. That's for another day. Well, that's an interesting story as to how cells make switch from carrying out glycolysis to be able to funnel glucose moieties into pentose phosphate pathway. Beautiful concept in biochemistry, but that's for another day. So this is the structural consideration that I would like you to be aware of. And now just let's take a look at how either NAD or NADP are involved in redox reactions. Keep in mind that NAD is used for catabolic reactions, and NADPH is used for anabolic reactions. It's very important considerations that you got to keep in mind. Okay, now I'm not going to explicitly give any example of redox reactions involving NADPH. You can look at it in any textbooks or research papers. In principle, whether it is oxidation or reduction reaction, when either NAD plus or NADP plus is involved as a cofactor, it is this pyridine moiety which is involved in, or nicotinamide moiety which is involved in redox reactions. The rest of the structure has got a role to play in ensuring that specific enzymes pick up specific cofactors. It's as simple as that, nothing more than that. So basically, if you look at any redox reactions where NADP is involved or NAD is involved, they would simply depict this part of the structure, and the rest of it, I'm talking about the rest of the molecule, all of this molecule is shown as an R group, okay? And I'm exactly going to use the same thing as an example to show how NAD is involved in redox reactions using the reaction catalyzed by lactate dehydrogenase as example, okay? Now what we have here is the reaction catalyzed by lactate dehydrogenase. Before that, let's just quickly take a look at how NAD participates in redox reactions. Okay, one second. I think I went back. Okay, fine. Okay, let's quickly take a look at that, and then we'll move over to the example of lactate dehydrogenase quickly, winding up this lecture. Okay, now what you have here is the oxidized form of NAD. Okay, now please keep in mind that this nitrogen is in cationic form, and the pi electrons are intact. Okay, the pi ring is intact, and please pay attention as to what happens when NAD receives 2 electrons. These 2 electrons can be coming from any intermediate in a catabolic reactions which is oxidized. Okay, it's as simple as that. So you're getting 2 electrons from an intermediate which is getting oxidized. Okay, it's as simple as that. Now what's going to happen is these 2 electrons, the pi system, are going to go to nitrogen, and nitrogen is going to become typically the one that has 3 bonds. Okay, 1, 2, and 3. Okay, and these 2 electrons are going to move here, and that will result in this carbon atom being slightly not slightly, being electron deficient, and therefore to compensate for that, these are all enzyme catalyzed reactions. So the entire active site of the enzyme is built for this kind of reactions to be facilitated. But to compensate for the fact that this carbon now has become electron deficient, a hydride ion is going to come. Okay. This is a covalent bond, single bond, which has 2 electrons in it. Okay, and these 2 electrons that are accounted for here. I mean, the 2 electrons which are going to be taken up by NAD molecule are going to go and form this bond with the hydrogen. Okay, so basically what comes into the reduction reaction, or I mean, if you're looking at the fate of NAD plus, it's reduction. But if you're looking at fate of the substrate which is getting oxidized, it's oxidation reaction. Okay, so basically 2 electrons are removed from an intermediate which is getting oxidized. And think about glyceraldehyde 3-phosphate dehydrogenase catalyzed reaction, right? You'll be able to figure out what's happening. Okay, so those 2 electrons along with the proton are going to be used up as hydride ion to form this additional bond. And now this nitrogen is no longer in cationic form. That's something you got to keep in mind. And this is the reduced form of NAD. Okay, this is the reduced form. It could be oxidized by complex 1 of mitochondrial complex of electron transport chain if this NADH molecule happens to be inside the mitochondrial matrix. Okay, something which is generated by enzymes of tricarboxylic acid cycle. If not, there is a shuttle system which is operating to move electrons from NADH from cytosolic domain to mitochondrial domain for eventual reduction of oxygen into water. Okay, it's as simple as that. So when 2 electrons are taken out of a substrate which is undergoing oxidation, those 2 electrons along with the proton are used to form this bond. Okay, so what comes into the moiety is a hydride ion. It's as simple as that. And the electron shifts happen so as to keep the valencies satisfied. Okay, so this is the reduced form. Okay, now we look at one reaction where NADH is used in the reduced form as the cofactor. Of course, the enzyme lactate dehydrogenase, which can catalyze reversible reaction, but we're only looking at the reaction in one direction, which is formation of lactate. Okay, what you have here is pyruvate. Okay, this pyruvate is going to be reduced to lactate using 2 electrons which come from NADH. Okay, so assume that this is the moiety which is going to be used as cofactor when lactate dehydrogenase interacts with pyruvate. Okay, it's as simple as that. So what's going to happen is now this NADH has got 2 electrons to be given away in the form of this hydride moiety. Okay, right? Correct. So where do those 2 electrons go? In a reaction which is catalyzed by the enzyme, okay, this bond is going to pick up a proton from the cytoplasm or wherever the reaction is taking place, and it's going to form this OH bond. Okay, right? So as far as these 2 electrons are concerned, we have accounted for where do these 2 electrons go. Okay, when these 2 electrons pick up a proton to form OH bond, this carbon will invariably be electron deficient. And to satisfy the valency of this carbon, 2 electrons from NADH and a proton are going to be used up as hydride ion to form this bond. And that gives you lactate. It's as simple as that. Okay, so what you're doing is you're converting pyruvate to lactate. And this is something that happens when your cellular ATP requirements far overwhelm mitochondrial capacities, or if there is an impairment in mitochondrial processes, you'll always see that a lot of pyruvate is reduced to lactate just to be able to generate NADH in the oxidized form, because unless you have cytosolic availability of NAD in the oxidized form, will not be able to carry out glycolysis. I mean, not be able to maintain the flux through glycolysis, because the enzyme glyceraldehyde 3-phosphate is going to choke down unless you can give it NAD in the oxidized form. Okay, that's for another video. But what you've done in today's video is you looked at NAD, NADP, the structural considerations of both the cofactors, what happens to NAD when it undergoes reduction, and one metabolic reaction where NADH is used as cofactor. If you like this video, kindly subscribe to my channel. It will only encourage me to make more videos related to biochemistry and post, so that you can learn from it. I can learn from interacting with you all. Thank you so much. It was a pleasure giving this lecture.